next group that's coming up to talk is Agilis Biotherapeutics. And I want to mention while they're getting mic'd up, when we started FAST back in 2008, one of the things we said, the board members would say to each other, is we're like, if we could just get a really good pharmaceutical or biotherapeutics company to look at us and see the promise in Angelman Syndrome, we'd be set. So, there's a culmination of a dream here today, having Ovid talk about a trial that's coming up and having some other companies that are going to come up and tell you about their approaches too. This is what we've always wanted. So, be happy you're sitting in the room today. This is a banner day. All right, so our next speakers are going to be Gregory Robinson and Jodi Cook from Agilis Biotherapeutics. Greg is the Chief Science Officer for Agilis. He brings more than 20 years of pharmaceutical and biotech experience in research, clinical development, and R&D management. He spent the last 7 years at Shire and then made the jump to Agilis. And he is a biochemist with degrees from Boston University and is here to tell us a little bit about how Agilis is doing what they're doing. Jodi is the Vice President of Operations and Strategic Alliances. She has over 20 years of senior executive experience in the life sciences. And she earned her Ph.D. in auditory science at Arizona State University, and that sounds really interesting, so I'm going to get her to tell me about that later. So we're going to turn it over to Agilis. Again, they're going to give an introduction about what they're doing, and then we're going to open it to questions from the audience, both from the live feed on Twitter and our Facebook page, and also in the audience walking up to the microphones. So please help me welcoming Agilis Biotherapeutics.
Great, thanks, Becky. And I'm really thrilled to be here to tell you about, Agilis's efforts to find a therapeutic for Angelman syndrome. I want to thank the organizers, Paula Evans, Allison, and everyone from FAST for inviting us here, and we're thrilled to be involved with this organization. I'd also like to put a special thanks out to Dr. Ed Weber and Dr. Kevin Nash for the efforts they put into Angelman and the work we're doing with them. It's working out very well, and I want to thank you guys for all the efforts. So I want to tell you a little about what's happening and what Agilis Therapeutics is all about. As Becky said, I've worked the last 7 to 8 years in the rare disease space for Shire Pharmaceutical, dealing with patient groups such as this, et cetera, and have really come to appreciate what it needs to go into the rare disease space to find a therapeutic. And because of the efforts in the growing momentum I'll talk about in gene therapy, I decided to make the jump and use the gene therapy to find therapeutics for genetic diseases such as Angelman and other ones that we're looking at in our pipeline. So on the next slide, do I? Thanks. Here we go. Is Agilis Therapeutics, we're really dedicated to advancing DNA therapeutics, innovative DNA therapeutics designed to provide efficacy in rare genetic disorders of the CNS. So we're concentrating on CNS, rare disorders, and how we can use DNA therapeutics, primarily gene therapy, to move forward and develop a viable therapeutic. We're doing this through, one of the more comprehensive scientific analysis of the gene construct, of the vector. I'll get into AAV, adeno-associated virus vectors that we're using. But we're looking at, synergistic platforms, looking at not only optimizing the DNA that we're using, the regulatory elements to drive that DNA and drive expression of the protein, but also the delivery platform using the very safe AAV, adeno-associated virus vectors. And then using different forms of delivery. We feel that direct delivery to the brain, direct delivery where you need to see the activity is right now the best way to move forward. So we're using direct delivery for the treatment of our therapeutics and our indications. So why gene therapy? And this was one of the reasons I made the jump to Agilis, is I didn't think gene therapy was moving fast enough at other pharmaceutical companies, and there's a lot of talk, but I wanted to see some action. And really, in gene therapy, there's been a dramatic increase. You've seen it in the news, but there have been clinical successes at Unicure, Bluebird, Spark, to name a few, as well as the environment around the regulatory environment with the orphan drug acts, the benefits you get from orphan drug for gene therapy, as well as the approval of Glybera in Europe for the first gene therapy approval. So there's been a lot of talk, but now a lot of action. Now there is a lot of action in the gene therapy space. Also, manufacturing was always a problem, but now that they've sort of solved a lot of the manufacturing issues and they've been able to deliver enough to make a therapeutic, that's also being dealt with and has really improved the chances of a therapeutic, gene therapy therapeutic being a viable option. And finally, you see significant value creation. And by this, I mean there's investment going into companies who are doing gene therapy. The Wall Street, the investors are realizing that this is a place that they can create a therapeutic, and because of that are putting money into it and allowing companies to form therapeutics. And as Becky stated earlier on, that the research going on in AS is going at a dramatic pace, a very increased pace, and we wanted to use the momentum of gene therapy to be there as well, and use gene therapy to the momentum that's been shown to create a DNA therapeutic for Angelman syndrome. So Agilis is really at the intersection of a lot of enabling capabilities. We have the scientific expertise. We're specializing in gene therapy. We have the expertise of scientists like Dr. Weber and Dr. Nash, as well as others, experience in the rare disease space. We have tremendous partners and advisors on our staff, who have experience in the rare disease space, have experience in synthetic biology, have experience with DNA vectors, with gene therapy. So we're able to move it forward efficiently and appropriately. We also, work with organizations such as yourself to move this forward. It's invaluable, the information you bring, the questions you ask, the passion that you have for getting a therapeutic, and that is very helpful for us. And also the progress in gene therapy. With all the things that have happened, it's not talk anymore, it can be action. And that's why we want to be involved and move our therapeutic forward. This is a quick snapshot of our pipeline where we have a number of different areas that we're working on. Angelman is one of the main areas that we're working on, along with Friedreich ataxia and another clinical stage that we're not announcing right now. But Angelman and the CNS disorders are ones that we're concentrating on and are prioritizing right now in our pipeline. So let me give a little tutorial on what is gene therapy. Everybody knows, it sounds very, sci-fi and all this, but really there's a defective gene. You need to replace that defective gene. So we find the gene. How do you get that gene into the cell that's needed? Right now there are viruses that everybody has in their body. They're safe, that you can use to deliver that to cells. And either you deliver it to the cell and then provide the cell to the body, or you provide it to the site of action, in this case the CNS, and allow the virus to transduce or move into the cell and then have expression of the normal protein, not the defective protein, or the missing protein in the case of Angelman syndrome. So really it's just a matter of getting protein expression through a DNA construct into the cell that's needed. We use a vector called adeno-associated virus, which is a very safe and well-known virus. It infects or transduces dividing and non-dividing cells and doesn't get integrated into the genome. It stays as an extra chromosomal piece of DNA that then expresses the protein of interest. So you don't worry about the integration into the genome and what might happen with that. The nice thing about this is there are multiple variants, serotypes of the adeno-associated virus that can target different cells. So we've been doing a lot of evaluation on the different viruses, different serotypes that can target the cells that we need for Angelman syndrome. Up to date, there have been no significant adverse events with any of the therapies that are using adeno-associated virus. So it's very safe, it's well tolerated, and it offers good cell targeting. So let's move into Angelman syndrome. This is somewhat of a rehash because Becky gave a great overview of Angelman and the causes of the disease, but essentially through imprinting, you essentially don't have the paternal allele, you have the maternal allele in the brain. In the body, you have both alleles being transcribed. But in the brain, you only have the maternal allele, and when that becomes deleted or mutated, you end up with a lack of UBE3A gene expression or E6AP protein being produced, and you get Angelman syndrome. So the molecular pathology of Angelman syndrome is well known. You know, you lose through deletion or mutation the UBE3A maternal allele, and therefore you lose the expression of the ubiquitin E6AP protein. And with the reduced expression of the UBE3A gene expression and E6AP protein expression, you end up with a decrease in neuroplasticity, a decrease in AMPA receptors, a decrease in the synapse efficiency and activity, and therefore impaired cognitive function that you see in the disease of Angelman syndrome. What's nice is that there's no morphologic pathology. In other words, as Becky was saying, in the adults, it's not that you don't have a heart or something like that. The cellular machinery is all there. You're just missing the protein that's needed. And the phenotype appears to be biochemical only. So if you can replace the protein, the E6AP protein, you should be able to restore function. So for gene therapy at Agilis Biotherapeutics, we've licensed with the University of South Florida, Drs. Weber and Nash, the worldwide exclusive rights to the Angelman syndrome gene therapy. The pioneering work that Ed Weber was able to do in the mouse model convinced us that and we felt gene therapy, but that showed the proof of concept that it would work in not only developing, but also in the adult animal, proving that there was no morphological insult, and you just had to replace the protein. And the work that he did was published in the daily paper in the PLOS ONE journal, and it showed that you could rescue the cognitive defects in a mouse model of Angelman syndrome. So gene therapy for Angelman syndrome, essentially, on the left-hand side of the slide, I show the UBE3A gene, which I've described, and an AAV capsid, a safe adenovirus. I should say that most everyone here has some serotype of adenovirus in their body already, so it's not something foreign to the body. Essentially, we'll be delivering it. This would be delivering to the CNS, looking at delivery through different mechanisms that you can get into the CNS tissues, either through the CSF or other mechanisms. And then those will be using the transduction or targeting ability of the adenovirus, be able to get to the cells that are needed for cognition, movement, seizures, things like this. So it's been in the work that Dr. Weber showed, he showed that the E6AP protein expression, the expression from the UBE3A gene, was normalized in the hippocampus. The hippocampus is needed for learning and memory. He was concentrating on the cognitive effects. He showed that synaptic plasticity was improved, that you could see the synapses were working, the neurons were working and functioning much better, and that he saw associative learning in the memory phenotype was recovered in these mice, showing that the E6AP protein was functional, had the ability to work in this model. So at Agilis, what we've been doing is working with Drs. Weber and Nash as collaborators, and have moved to the human UBE3A rather than the mouse UBE3A. So making a therapeutic with the human UBE3A gene construct. And we've created the human UBE3A construct. We're working to optimize different portions regulatory to get the optimal expression of the E6AP protein. And I show the construct here with the different elements that we're using. And right now, we're actively working with both labs and looking at testing different AAV capsids to find out which ones deliver. We're looking at evaluations. We'll be using the AS mouse model, the similar model, but expanding on it. Using the human gene, but expanding on looking at how you can more broadly affect the different parts, such as seizures, such as movement disorders. But looking at learning, memory, neuroplasticity, right now in the lab we're doing all these experiments, and they are ongoing as we speak. So we've started the work. And hopefully, we plan to have a lead candidate selection of a gene therapeutic that can go into clinical development in 2016. So I'd like to end the science part of the talk now and turn it over to Jodi Cook, my colleague, who will talk more about the development of this lead candidate into a therapeutic. So thank you.
Okay, great, thank you. Thanks, Greg. Thank you very much. As Greg said, he runs the science portion of the company. We're a small company, so we're all very actively involved in everything. And he said he'll have a lead candidate by 2016. Remember, we just licensed this and kicked off the work in July with Dr. Weaver and Dr. Nash. And I told him, I was like, 2016, that's not fast enough. Can't we have it by Christmas? So I'm with you. We have to do it right. We have to be really focused and make sure we make everything correctly. But it's never fast enough for all of us. I understand that. Let me tell you a little bit about the drug development process. This is probably relatively new to all. What's new to all of you? It's certainly newer to people that have never done drug development. You've heard before that maybe 1 out of 10 out of 10,000 actually get through into the process. So we're a little bit ahead of the game in that by licensing the technology and the great work that had been done by Dr. Weber. Agilis came into this process beyond the discovery phase. So we came into this at the preclinical phase. July of this year, we started in the preclinical phase. And two things you have to do together. You're doing the preclinical phase of the work. What does that mean? That means he proved it in mice. Now we need to make it for a human. So they made the human version of it. And now we need to retest that version a bunch of different ways. Is it safe? Is there a mistake? We look at it in cells. We look at it in animals. We look at it in various ways. So that's all the preclinical work that's going on. The other thing we have to start working on right now is something called process development. When you actually have a pharmaceutical that's approved for use, you have to show that you can make the same thing over and over and over again. You can make it in a laboratory, but then you have to be able to produce it so patients all over the world get the same product. That takes several years of development. So we've already started on the process development. So what comes out of the laboratory then needs to have a lot of controls put around it so that we can make the same thing over and over again. So that goes on behind the scenes while we're moving things forward. Once we get to the point where we believe that the scientists have come up with the right candidate, we believe we understand how to make this, then we'll engage with the regulators and talk about essentially what's called a pre-IND meeting, where we say, okay, we believe this is going to work. Now we discuss with them, what do we have to do? We have to do a series of safety studies and a whole series of other things to make sure that what we're going to put into a human is safe and can be effective. So we have those conversations. Once we have those conversations, agree to the plan, you execute on the plan, and at that point you're able to start your phase 1 clinical trial. Phase 1 clinical trials are safety studies. Generally, they work with some dose escalation, so you do a little bit of dosing in that, but they're really designed to make sure that what you've developed is safe. Everybody looks for efficacy signs, but it truly is a safety study. So once you get into phase 1 and have demonstrated it's safe, then you go back to the agency and discuss with them how to take it to the next level, how to broaden the population, how to show that it is effective and how it can work. So it really is typically a 7 to 10 year process to get from an idea through into a phase 3 and proof that it's available on the market. And honestly, probably 7 to 10 years in general is optimistic or pushing it a little bit, but it takes a while to get beyond just the idea phase. So that's the process that companies go through. Where we are, like I said, in Agilis, we're at the preclinical stage. We're working aggressively, committed with Dr. Weber to get us moving to the next round of studies and the process development. We hope to have the lead identified this year, this year coming up in 2016. At that point, we will launch into those pre-IND, IND-enabling studies. Many of you have, we announced that we had received orphan drug status, and many of you have been reaching out to us and sending us emails trying to understand what that means. The Orphan Drug Act was something that was passed in 1983 that was designed to promote development of orphan drugs, orphan indications. So for diseases that have fewer than 200,000 in the United States, that act put into place economic incentives to try to get companies or biotech companies to develop these products. There are a number of economic incentives there to help us do this at a lower cost. Since that has been put up, and the other thing that, that goes along with this is an ability to have the companies interact with the agency more often or at a faster pace. So for a company like Agilis, that's really important. We don't want to launch a year's worth of studies, go talk to the agency and find out, oh gee, they really wish you had done this. So we want to interact with them as much as we can early on, design the studies together so that we don't make a mistake along the way, so we don't waste any time. So we're very focused and committed to making sure that before we take the step, before we do the study or spend the money, that it really is the best path forward. And that is what the Orphan Drug Act for us has been a big benefit, will be a big benefit. Excuse me. In general, since the Orphan Drug Act has been passed, there have been over 3,000 approvals, and nearly 500 orphan drugs have been approved for sale. That's an enormous increase from before the act was passed. There were 10, and now there are over 500. So this has really opened the eyes to the agencies, organizations like NORD or your organization. Plenty of organizations, rare disease groups are combining and really making an impact into the legislatures, as well as bringing awareness to the public. There are thousands of orphan diseases, and together that's a very large number. So there's a lot of companies working on this. So, did it switch? So for Agilis, we announced this. We were granted this designation in October of this year. It'll provide financial incentives to develop our treatment for Angelman Syndrome. And really important for us, it'll provide more frequent interactions with the regulators and potentially speed our development and our entry into clinical trials. And that's a really important thing for our company. So our next steps for Agilis, as Greg had mentioned, we are working in the laboratory to optimize our DNA and delivery. Dr. Weber showed how to do it in a mouse. We're looking at, are there other ways, this piece of DNA, can we put different things around it to target more specific cells? Your scientists have done a great job identifying what's the problem. So now if we can just tweak what we have already to maximize the potential benefit, that's what we're doing in the laboratory right now. That should be completed in 2016. And then we'll move into what's called safety studies, where we will dose at higher and higher levels in an animal model and determine what we believe will be a safe dose for the human. And obviously the goal is to give the safest dose that will give the most benefit. A safety study could take 5 months, it could take a year. Again, it depends on what the results show up until that point and how we have that conversation with the regulators. And then we are going on with the manufacturing development. There are a number of different places that have, over the last decade, worked out how to manufacture gene therapies. These are biologics. It's not quite as simple as just making a pill over and over. There's a lot of reaction going on in making these products. So it's taken a while, but now many companies have worked out how to do that. So now we need to modify that process for this specific one for the treatment of Angelman syndrome. We are also concurrently in the areas of trying to understand what's the best route of administration for the gene therapy product. The disorder that we understand is mostly in the central nervous system in the brain. So we're looking at different routes of administration. We're talking with neurosurgeons, neurologists, the scientists in this room to help us understand what is going to be the best way to deliver this to your children and family members. Is it going to be maybe like an epidural, a lumbar puncture into the CSF, cerebral spinal fluid? Is there other ways that we can deliver this that will provide the most efficacy? We are also looking at what should the phase 1 clinical protocol look like. And I know that's not coming right away, but we need to understand what to do and what's the best thing to do. There needs to be outcome measures to evaluate improvement. And you heard about that in the previous talk as well. What are the right things to measure? A lot of clinical trials end up failing because they didn't measure the right outcome, or they didn't know what the right outcome was to measure. We want to make sure that we start today to figure that out so that we at least de-risk that as much as possible. And I'm going to talk about that a little bit more in just a minute. We have going to engage regulatory discussions, and then you open something called an IND, which then allows us to move into human trials, and then obviously then into a phase 1 human clinical trial. So let me just tell you a little bit about something that I'm very excited about and have really been diving into the last few months. I'm calling it the Outcome Consortium Mission. For the Angelman groups, the Angelman syndrome, we need to establish reliable markers, clinical measures of Angelman syndrome that can be used for therapeutic development, evaluation, and registration. That's different really from a test that you would go to your doctor and get, or an educational evaluation to put your child into special services or get extra help. This is really something that needs to be measurable and repeatable, and you can put statistics around so that when companies, whether it's Agilis or any company, goes in to say, we have a treatment, we believe it works, it can't just be because the parents say it, the kids are better, or we think it's better. We have to show scientific evidence that it works. So we need very clear outcome measures, biomarkers to do that. So Angelman syndrome as a group needs to have those for whether it's us or any other company down the road over time. You're going to need to have those measures. And we've already heard about it in the first talk that they're looking at what are the exact measures that we need to use to find an outcome that can get a therapeutic registered. So the goal of this consortium mission would be to invite all stakeholders to participate in the project development. I've already been to, thanks to Terry Jo at Vanderbilt, we've been to Vanderbilt. We've had a group of scientists together. We've been to South Florida. We've seen Ron Thiebert in Boston. We're going to see Ben Philpott and the UNC team next week. So I'm starting by traveling the country, and I've been talking to the folks in Australia. Megan, I've talked to her several times as well. So we're really trying to get all the various stakeholders, the clinicians, the scientists, to at least open their eyes to, and they've all thought about it individually as I go around and talk to them, that these are really important things. So we want to get everybody excited about it and then pull them all together and convene a planning meeting. So hopefully early next year, we're going to convene a planning meeting to discuss this short-term and long-term outcome measures. So we need some that maybe for those of us that are entering clinical trials in the next few years, and then maybe there's a global Angelman syndrome outcome measure that gets developed for something that enters clinical trials in 10 years. But we need to raise awareness, get participation, get funding around this, and then execute this plan and continually update on what those measures are. Those are the things that can happen right now, today, so that when any of our therapeutics enter into registration process or talk to any of the regulators, they've already been validated. They've been validated by the community, by the scientists, by the clinicians, and so it's much easier. It will speed the process for any of us to get therapeutics. Some of this has already been started, but certainly we'll want to rank order symptoms that are important to families and caretakers and clinicians, and I know you've already been started on doing some of that, but really to develop effective therapeutics, it requires the families and the patients' interactions, because you know your disease better than anybody. Even the clinicians and scientists, we may say, this is what we think is highly important, but then we have to hear from you. And so if we do drug development work that's inclusive of the family and patients, you end up with a therapeutic that is much more targeted to the big needs of the community. So all stakeholders need to be involved. We need to find those sensitive objective measures. If they're not there, we need to identify them, plan and budget, and then we can raise funding and execute the plan. And you've done a fantastic job of already moving things forward, but this is something else I think that we can work on. I've already had conversations with people at the NIH that are very interested in funding and helping to make these programs move forward, and they're very excited because I tell them that there are very active patient family groups that are part of that, and those are the kinds of things that they enjoy moving forward and helping to fund. So I just wanted to have a few, I put in a slide here called Common Questions. Many of you have emailed me, and I look forward to actually meeting you, and thank you very much for sharing your stories and your photos of your beautiful children. And probably the top 3 questions I'm getting right now are, will your child qualify for clinical trials? We don't know yet at this point what the inclusion-exclusion criteria are, but like all clinical trials, you start with something. You try to make it as tight as possible because you're looking for getting the first group through that gives you a good result. But that means that very quickly behind that, you follow up with broadening those inclusion-exclusion criteria. So we are not planning to ever exclude anybody. The therapeutic idea is to treat the entire population with AS. When will it be available? Again, I really can't answer that question. I wish I could. I can give you the near-term timeline, which we've talked about. We hope that our science will develop the lead candidate next year, and then we will begin the conversations with the regulators to get to what are the right set of studies that we need to execute. And then is there anything I can do right now? And really, I think you're already doing all great stuff. If you can, I was talking with Megan in Australia, and I don't know when that's going to happen, but in the very near term, they're launching a registry, a patient registry, which I think will be fantastic. So join that when that happens. Keep very good medical records if you can. If you haven't had the confirming test, the diagnosis, you can maintain or get that. And participate in anything that you can to keep things moving along. Any therapeutic that comes on the market is not going to immediately, nothing is going to snap your fingers and be everything's perfect. So if you're in therapy now, keep that going. Very likely, even with a gene therapy, you'll have improved function, but you're still going to need therapy to teach and make progress from that. So nothing is going to be a snap and it's all done. So I would say keep going. You're doing phenomenal right now. Keep up what you're doing, and we will keep you posted along the way as things move forward. My last slide here is just a picture of our team. Like I said, we are a small team at Agilis. We are 3 full-time employees, and the way that our model was set up is to be very collaborative. We have an excellent group of scientific advisors, and we use them regularly. We have Dr. Weber, the group at South Florida. We have the FAST organization. And one way that the Agilis investors decide when they're going to take on a new disease or have something for us to work on is there enough support in the community to make this happen? And you should be very proud of yourselves, because the answer is absolutely yes in Angelman. And that was a big reason. Dr. Weber's science is outstanding, but if this group didn't exist, it's likely we would not have moved into this space. So you guys are phenomenal for doing that and for being active. When we were investigating whether or not to take on Angelman syndrome, I sent an email to Paula, and I happened to be in Chicago for personal reasons. And she's like, "Okay, I'll meet you. Where are you? I'll come talk to you." And now that I know Paula, as everyone else does, once she's got you in, there's no going back. So thank you, Paula, for meeting you and for getting us all excited and interested. But I can promise you that we're a small team, but we're a very committed team. And we're committed to rare diseases. Friedreich's ataxia was our first indication. We joined them for many of the same reasons as this. And you guys actually were our second indication we picked up. And quite honestly, this one has moved quite fast. And I promise you that we will keep it going as fast as humanly possible. And that is because of all your input. So thank you for having us here. Thank you for welcoming us into your group. Sorry, I have a... okay, so we can open it up to questions. I don't have any.
Okay, again, we were going to ask you to come to the microphones.
Why don't you just stand up?
And
Yeah.
Also, we had said if you're on the live feed, you can tweet us or you can go to the Facebook page. There's also a conversation going on the live feed page. You can ask questions there as well. So we'll start over here.
Hi. Good morning. My name's Colin. And really, I've got a funding question for you and for Dr. Levin, really, just to give us an idea of the amount of money that you actually think you're going to need over the next 7 to 10 years to get to the point where you can actually deliver a drug. So have you got it? I assume not, because you were talking about funding becoming available during the process. And then for Dr. Levin, I assume you're on a shorter track, therefore need less money. But again, it would be helpful if we could hear from you the sort of rough quantum of funding that you're going to need.
Yeah, have him go into it, because he's shorter track. And he's brought more drugs to market than I have.
So I have a principle. Don't ask other people to put your money into a company unless you put your own money in.
Fair enough.
That's number one. So we did. Me and my family put in $5 million last year at the beginning in August, and friends and others. And then in August of this year, and Matthew as well, the management of Ovid invests in its own company. That's number one. Number two, that was just the beginning. That was to prove that we could actually collect and be ready to help people. And in August of this year, we raised about $75 million. So we have more than adequate money to bring this drug to the marketplace.
Okay.
And hopefully we'll do that successfully. But again, in the August fundraising, the same principle. Put your own money up. Don't ask anybody to put other money in. And the money we have is not venture capital. It's not quick money. It's not money that wants in and out. I picked it very carefully. These are people who really believe in medicines. They must meet the philosophy that we at Ovid have. And if those of you care to visit us in New York, and I think, Alison, you have, and I know that you have, you'll know that we don't live in fancy offices. Yes, I have a tie and a jacket and a suit on today, but that's out of respect for you. I am, without a doubt, the oldest person in our building. We live in a WeWorks. WeWorks, for those who don't know it, is we have everybody crammed together, 6 in a room. My desk is that size, and most of the people in there are entrepreneurs. So we're investing our money in this, nothing but this.
Okay,
So-
that's helpful. Thank you.
Yeah. The Agilis team was started, actually it was a purposely formed company in synthetic biology, in particular gene therapy, by a group of family investors, mostly, that had a mission of using synthetic biology to do something very meaningful. And they worked with a company called Intrexon Corporation, and R.J. Kirk has done a number of very successful biotechs, and put something together in the very beginning with some key investors and who are on our board of directors right now that are committed to doing this. Financially, where we are is that we have enough money to get us to major milestones. And in all biotech drug development companies, you don't necessarily pull it all in the very beginning. And as you make progress, you bring more money in. The current investors are highly committed to this, personally committed to this, and will continue to put their money into moving things forward. So I'm honestly, we were talking about this the other day, I'm not really worried at all about making the next round of funding when that becomes necessary. But we are, and like you mentioned, ultimately it takes a lot of money to bring a drug to market, particularly from the very beginning. But as we make progress and as we see positive signs, the money is very much there and available.
So
Okay, thank you.
I just want to say from the community standpoint too that Jodi took you very nicely through the process of getting a drug to trial. What we're doing when we're asking you guys to continue helping us raise money and things like that for FAST and other organizations is we're trying to fund the basic research that's going to provide those preclinical data, that's going to provide those targets that then the pharmaceutical companies can take through their pipelines. So I don't think anyone in this room needs to worry about raising money for the companies. They can do that on their own. They have their own ways to do that, and they have a lot of places to go for that we don't even have access to. So we're not going to rest until we have multiple therapeutics for Angelman syndrome so that every single kid has something that works for them. The last thing we want is to have something that works really well and 10% of our kids are allergic to it or can't take it or whatever. So we're going with the basic research. We're still looking for all the things we can do so the pharmaceutical companies have something to do with it. So that's just my point for this.
And can I just make one more comment on that? This is in my view, in our view, this is a pre-competitive space. We're not in competition with Ovid at all and other companies. It's not, there are no indications where one company gets one treatment and that's it. It will take a number of different, maybe a cocktail of treatments or various varieties of treatments from multiple companies to cure this problem. So this is not, there's not, oh, I like that company or not that company. We're not in competition. At this point, we're looking for the first therapeutic and then the second, and it's going to be a lot of therapeutics before we get to where we need to be. Go ahead, thanks.
Hi, my name is Sharon, and I want to thank you for investing in the hope we have for our children. My question is this: you mentioned that you've achieved long-term expression in non-dividing cells and wanting to optimize the levels, and obviously the initial part is trying to get enough expression in all the areas that it needs to occur. But I'm wondering if you're also looking at regulation for overexpression.
Right now that we're looking for optimal expression, so as you said, in the different cell types. For overexpression, in vivo, a lot of times, you have to see, do you really get enough expression? And that's the key. We are looking at different ways, though, and have discussed, what are called mammalian switches and things like this if you need to lower the level or remove the gene or whatever, a safety switch of some sort. We are discussing that. Right now, our primary goal is to get something that expresses enough.
Thank you.
Hi, my name is Kathy, and my question's kind of elementary. In your presentation, you referenced E6AP or something like that, and all the stuff that I've read talks about UBE3A creating a UBE3A protein, so I kind of wanted to understand the difference between that and the one you talked about.
There's no difference. E6AP is the UBE3A.
Oh, okay.
The protein that's produced from the UBE3A gene.
It's just a different name?
It just has another name because of how it was discovered.
Okay, thank you.
Hi, my name is Anna. My son Aiden is 10. My question has to do with when I listen to these two talks, I have a hard time kind of integrating what's going on. So I understand your work is further upstream and the OV 101 work is further downstream, but there are words in terms of learning and potential, and then there are words in terms of tonic inhibition. So how do these two efforts kind of come together?
We're trying to fix the broken gene, essentially, and it would be we're replacing what was broken so that the expression of the protein occurs and all the natural functions come back. So we're at the level of the gene. Let's fix the gene, and then all the natural functions should come back. Versus or in a different, yeah. I don't know what to say. Sorry, I won't speak for you.
So yeah, it's a great question. We believe that if your child had been born with a healthy copy, without the deletion, without mutation, it would be a child without Angelman. I think that's the understanding we all have. And so the hope ultimately would be if that you go in and intervene and put the healthy copy in at the time of conception. Remember, it's important. We don't know from certain animal studies, we may not be able to actually get 100% recovery. So as a physician, as a practicing physician, we never use the word cure in medicine. In fact, that's something that we're all dying, medicine advances through incremental mechanisms, right? We make small leaps, and those ultimately add up to something that's really revolutionary. But there's very few changes in medicine that are paradigm shifting, that are totally disruptive, that cure. So we're looking for incremental steps. Ultimately, the approach that Agilis is taking, they would ideally like to be able to intervene in utero, right at the time or early on conception. We now have tests that, in fact, we can isolate fetal DNA from the blood, and we can screen for Angelman, of certain, the microdeletions at least, during pregnancy. So that suggests that you have the opportunity, as science advances, to intervene at that earliest stage, even in utero. And that's the hope in the future of gene therapy and other genetic approaches, gene editing, other approaches that can create recovery. Our approach right now is to say, every one of you has a child, or those of you in the Angelman community, has a child who could be helped. If we have a medicine that we believe can help you at some level, we want to get that to you safely and effectively, and we want to make a difference in your lives. And small gains in many symptoms can translate to enormous difference in terms of quality of life. So that's our strategy now. We're very interested in the biology of this disease. We would like to make revolutionary changes. We'd like to be able to talk about, use words like cure paradigm. But right now, I think the goal for us to say, we want to be able to help you, we want to do it well, and we believe we have a drug that can do it downstream of the gene. In other words, as you mutate that gene, multiple things downstream change. We have seizures, not simply because of that. And so the goal here is to say, we have a drug that we think can help you, would like to get it to the clinic and do it fast.
So just
if I may make just a broader comment. So having seen many therapeutic areas develop, one of the most important things is to take note of what you said earlier on, is that many drugs are complementary to begin with. And the voyage that you take here is a voyage of understanding the biology, understanding truly, we think we know the answer, we may not. Sometimes even in the most extraordinary areas, we thought that we knew how to cure infectious diseases. In fact, the pharmaceutical industry was so arrogant that it decided in approximately 1980 to stop research in antibiotics. We've cured it, it's all done. Nonsense. Bugs are really smart, and in fact they've now come back. So as we look at Angelman's, I think what I can truly say is that every single approach that we adopt gives us more knowledge, number one. Number two, yes, there is a period of time. I don't know how long the trials are that take for gene therapy, and I don't know truly at this stage. I will come back to you next year and tell you exactly how long our trial are, because yes, we are much further ahead. However, it's my firm belief that these therapies will be complementary. It's my firm belief that as we go into it and we learn from your children, that there will be lots more to be learned downstream. And during that period of time, we also hold the hope, and this is what I really believe is important, is that somewhere along the line, we'll catalyze just a tremendous amount of new thinking about what are the basic disorder, what are the ways of approaching this. There were many proteins that Becky showed on that pathway. I'm not looking for a magic bullet, I'm looking for a medicine. And we need to be open to the fact that there are different approaches to that. And I can assure you, I'm certain that I know we are, and I'm certain that all other companies entering into this are doing, and if they aren't, beware.
Thank you.
Definitely, I want to second that, Jeremy, that absolutely, this is to cure Angelman will take more than one therapeutic. There's going to be differences. We don't know all the biology, so we want to make sure that we have enough therapeutics to move the therapeutic benefit forward.
Laurie?
Good morning. My name is Lori, and I have an 11-year-old son named Josh with Angelman Syndrome deletion positive. I never thought I would try to immerse myself in my leisure time in science before him. So if this sounds, on December 1st, I was reading an article from BBC News on the difference between gene editing and gene therapy. And one of the things that they said is that there's no way for scientists to know where the gene will end up in gene therapy when they send it in with the vector. So does that potentially mean that if you sent in the vector with the gene therapy, that it could be different in every patient? Or is it kind of like the majority of the patients, it would go to the right place, and some of the patients, it wouldn't? I guess I don't understand how that works if there's no way to know where that gene is going to end up.
That's a good question. That's one of the reasons, when I talked about integrating and non-integrating, when you integrate into the genome, there are specific sites, but you're not really sure where, and that was an issue. With the adeno-associated virus, you're looking at getting it to the correct cell. It will stay in that cell and produce protein, but it won't integrate. It's been engineered so it's safer and won't integrate. Okay. What you have to worry about is the cell that it gets to, and that's where we're looking at the what's called tropism or cell targeting to make sure you can get the piece of DNA into the cell so it'll use the machinery to produce the protein but not integrate into the genome.
Okay. And a lot of that depends on how you deliver your therapeutic as well. And like Greg said, these are non-integrating. And a lot of work that we've done even before we joined into this community was what type of viruses target what cells in what part of the body. There are some viruses that are really good for the liver. We don't want to be in the liver. There are some viruses that are better for different parts of the brain. So the scientists over the last 20 years really have been working out which viruses target which cells. So as we select that, then we do in a very, and Kevin Nash is doing a lot of it, a very exhaustive evaluation of once we've given the therapeutic, where are we sure it hasn't gone to the wrong places? So before we get to a human, we will feel very confident in animal studies that it absolutely targets the right place, but not the wrong place.
So is it typical then that if you've tested it and in the testing you've done, it's going to a specific cell per se, that it would generally most likely do that in everybody once you're realizing where it's going, that it's most typical that that would happen that way?
Yes, that's what we believe, yes. Yeah.
Thank you.
Okay, we're quickly running out of time, so these are going to be the last two in the room, and I have one from online from our live feed. With your streamlined approach, is there any estimated time frame on when phase 1 trials could possibly start? Give us your worst case scenario.
It is live streaming, so I'm sure that our board might be online, so I can't throw a date out there that I'll be held to. So, like I said, it's 2016. If we have the candidate targeted 2016, if we say safety studies will take another year, then it's developing the work, and then we could move into humans. So a couple years would be a reasonable estimation without putting a firm stake in the ground. But every time we talk to you, every time we interact, every time we will give you our next best guess. So our next best guess right now is that the lead candidate will be next year.
You've been standing longer, so why don't you go first?
My name is Stephanie, and I was just, I don't know exactly how to ask this question, but my son is Leo, he's 8, he has a loss, and he also has a gain. Now, is this gonna affect him being a part of any trial because he has that gain in his genes as well?
You know where the gain is?
Yes, extra X chromosome.
Yeah, we have quite a few. Her son is not the only child that we have in our community that has been doubly genetically blessed with Angelman syndrome and something else. Do you have some thoughts on whether that becomes an exclusion criteria for a trial?
As far as Angelman, I wouldn't think so, but we need to be, fairly stringent on what the initial inclusion criteria will be. But in general, I wouldn't think it would have any effect on a treatment.
Okay, thank you.
It'll probably be dependent on the trial and the drug, but I'm in the same boat with you.
But in the end, I don't think it'll make any difference.
And Stephanie.
Hi, my name is Stephanie. I also have a child with Angelman who's 11. But the first thing I have is a comment. 10 years ago, we thought, we've got kids with Angelman. What are we going to do? How are we going to survive? And never would we thought we would be here 10 years later, let alone here with 2 companies that not only honor 3 companies that honored the science and the human aspect of all of this. I remember talking with Dr. Weaver almost 5 years ago about all of this, about how we wanted our kids to be treated as human beings and not, unfortunately, the mice that we're studying. And I just want to thank you for respecting that, because it's very important to us that our kids remain our children and that innocence remains and they don't become a science experiment. So thank you for taking that under consideration. Having said that, researching the data, are you planning on utilizing data that's already been measured in other trials or in other research mechanisms, or are you looking at trying to find new metrics that are specific to Angelman syndrome so we can better evaluate the therapeutic that is being put to clinical trial, something that's a little bit better than the Bailey scales, for example?
Yes and yes. Yes, we are absolutely going to look at all the data that's been out there. In fact, we have just engaged with Dr. Bird and Dr. Hahn to accelerate pulling all the natural history data into a usable, queryable database that will be available to any company that needs to do research. So yes, you should never throw away old data. Always use it as much as you can. As we get smarter and understand better questions to ask, we can go back to that data that will be in a database and query it again. So absolutely, we're going to use that. Any studies that have been published with the data, we want to use that as well. The Baileys and the things that have been used over time, for sure, we will use that data. And then, yes, on the side of, should we develop more? When we were at Vanderbilt, we were interacting with circadian rhythm scientists. We were actually interacting with scientists in the auditory side, which is my background, for various evoked potentials for hearing and background noise. And so we're trying to borrow from other areas, other scientific areas that also Angelman kids have disorders or have symptoms of. So we don't have to recreate a new test. And then finally, I know Dr. Weaver's going to talk about it later today, I think. We've been talking with him as well about what is the right scale that really identifies the Angelman syndrome as a whole. And maybe that's a brand new development effort. And I think that's really important as well. So yes, yes, and yes. We're going to use old data, medium term. So as therapeutics progress through this, there will be needs for all those different scales in all the different areas. So we're trying to lead that effort to get it organized, to make it move faster. But certainly you guys are going to pick that up, and the scientists and clinicians will pick it up and run with it, so that when any of us end up in front of the agency, we have not only the idea that everyone says, yeah, we have this problem, but here are a number of different scales to prove it.
Excellent.
Thank you.
very much.
Thank you for your comment as well.
All right, let's thank
Thank you,
the
everyone.
people from Agilis. And we are miraculously on time. So we have a 15